Hi friends, welcome to YouTube channel Green Treasures. Today's video tutorial will cover the range of thallus structure in algae. As you know, the range of thallus structures in algae is incredibly diverse, ranging from simple single-celled forms to a complex multicellular structure. In this video, we will see the different types of the thallus and try to understand their complexity and their nature also. As you know, thallus organization in algae, it is having a diversity, and algae are diverse thalloid plants. Thallus is basically a vegetative body of the algae, and it serves as a main site for its photosynthesis, for nutrient absorption, for reproduction. But these organisms, fascinating organisms, being very simple, they lack advanced cellular and organ differentiation. That means we will not find roots, stem and leaves in these types of the organisms. So algae, as we have seen, it represents the most diverse kingdom as per its form and structure is concerned, and they show a great diversity in thallus structure. It will range from simple single cell to highly organized ones. And we have multicellular algae. They show differentiation of tissue, but that specialized tissues are meant for photosynthesis, support and reproduction. Mind you, they lack vascular system. That means these organisms does not have xylem and phloem. In case of multicellular forms, they look like filaments, sheets, blades and hollow tubes. So multicellularity is not highly advanced in these types of the organisms. You will see multicellularity in kelps and in seaweeds. As per as the size is also concerned, the size ranges from few microns to several meters. So that means here we will discuss the range of the thallus structure, which you might have understood is from a very simple single cell multicellular organism. Here is a summary chart where you will see the range of thallus structure. It can be unicellular, it can be multicellular. Unicellular can be a motile one, it can be non-motile one. So the multicellular can also be non-motile, it can be motile. And depending upon, we have different forms in case of these multicellular organisms. And then we have a filamentous type, we have a heterotrichous type, where there can be unbranched, there can be simple branched, there can be false or the pseudo branched, or in heterotrichous habit where you will find the prostate system and the erect system, they can be of these types, and there can be a parenchymatous type of the thallus, or there can be a pseudoparenchymatous types of the thallus. This summary chart we will discuss one by one. This will be our point of discussion in the next slides. So first of all, broad categories. There are 6 broad categories of the thallus in algae. One is unicellular thallus, the second is colonial thallus, filamentous thallus, and siphonaceous thallus, and parenchymatous thallus, and pseudoparenchymatous thallus. Let us discuss them one by one. We have a unicellular thallus that can be an acellular, a single cell plant body. Unicellular are common in all groups of algae, but there are exceptions. In Phaeophyceae, Rhodophyceae, and Chaurophyceae, they does not have the single cells, they have multicellular cells. So within these, there can be subtypes on the basis of the movement. That is, it can be unicellular but motile form. it can be unicellular but not non-motile forms. So we'll see unicellular motile forms. It is basically a single cell with a flagella, because the movement can be with the flagella also. So movement either due to flagella, or there are other protoplasmic projections which you name as rhizopodia or rhizopodium or rhizopodial projections. And movement is due to the flagella. A true flagella is here, which has a 9+2. We'll discuss it in the next slide, just to give a hint about it. But in the next video, you'll see the detailed structure of the flagella. And flagella attached at the anterior end of the body or the thallus. And it can be a whiplash type or tinsel type. Mostly whiplash type. Whiplash means here it is the flagella where we have an bend ends, wibs back and forth. So you have an unbranched with acute bend at the end. That is called as a whiplash. It is naked. It does not have the side growths. So the bend end whips back and forth. This is this end which bends back and forth and which helps in the movement of the organism. And number of flagella also vary in different forms of the algae. On that there is a classification we will see when we will discuss the structure and the types of the number of the flagella in different types of the algae. So here is a brief gist of how whiplash type appears, a naked type of the flagella where the tips will show to and fro here, and here is the filaments. Filaments is here. There's the side branches are here. This is called a tinsel. And when you see the anatomy of it internally, it has a central two elements and surrounded by means of the doublets 9. That's 9+2. And this structure will be discussed in the next video. Here we were concerned about the whiplash type of the flagella or the tinsel type of the flagella. Rest internal structure is same, 9+2. So unicellular forms may be spherical, oblong, pear-shaped. So as you see the Chlamydomonas, this is a pear-shaped. Here is that the flagellae are here, which is at the anterior end of it. And this flagella, when you see the internal structure of this pyriform type of Chlamydomonas, you find it has the largest this cup-shaped chloroplast. This is its nucleus, and this is the chloroplast, and this is the nucleus, and then this is the eye spot. Pyrenoids are present there. Contractile vacuole is there. So it has a flagellated unicells, resemble motile gametes and zoospores. Because you will find in some algae their reproduction is through the gametes. They also have the flagella. And here we have quoted the example of the Chlamydomonas, which is a single this motile type of an algae having a cup-shaped chloroplast and also the two flagella which moves in and help us in the movement of the organism. There is also one more organism which is the Eugulina, which has a one flagella here. It is the structure of the Eugulina. And in addition to this, you have Ameboid Forms, where the movement is not because of the flagella. They lack rigid cell walls. They lack flagella. And then how they move? They move by means of these protoplasmic projections, which are called as rhizopodial or rhizopodium. And they help in the ameboid movements. As you will see, the example is this is the Chrysameba species, which is moving by means of these ameboid like these rhizopodial type structures. And there is also Rhizocoloris belonging to Xanthophyceae. These are the motile forms, but they lack flagella. Their movement is because of rhizopodium. But there are also present the non-motile forms. Non-motile forms are a single non-motile cell because they are single-celled, carrying out all essential functions of life. They lack flagella. So here it is a single-celled but without flagella. You have a chloroplast there, you have a pyrenoid there, a nucleus there, eye spot is there, but here it does not have the flagella. They are non-motile forms. You have the chlorella example, and diatoms are there, and clostridium are there. So many examples are there which are non-motile but are single-celled unicellular structures. Then comes the colony type, colonial type, multicellular type. Colony formed by loose aggregation of individual cells. So here is a mixture of number of the cells. That is, numerous cells come together and they are embedded in a gelatinous matrix and they form a colony-like structure. And you know, cells usually connected with each other by cytoplasmic threads, and they will move as a unit. Each cell is capable of doing life processes. Individual cells are capable of doing the life processes, and but they move as a movement. For their movement is as a unit. That's called as the colony type of that's the colonial type. And we have subtypes in this type. That is, they can be motile Coenobial colony, non-motile Coenobial colony. There can be palmilide forms. There can be dendroid form. And so we'll discuss them one by one. Motile Coenobial colony, we have an example of volvox which you have already gone through. Definite number of motile cells are there. They are just like number of chlamydomonas type of cells, but in an aggregation, embedded in a gelatinous matrix, and they are connected with cytoplasmic connections, and they have a flagella all around protruded out, and which behaves as a single spherical motile colony. And the coenobium, a colony with definite number of cells having a constant shape and size. Spherical numbers, and numbers will vary, but the shape will be spherical. Whole coenobium move by the organized beating action of flagella. There can be a volvox which has 500 to 50,000 cells. Example is volvox. Gonium is there, Ponderina is there, and there are other species also. So that means we are reaching to from a single cell to a number of cells, but they are aggregations. Here is how Eugulina, this volvox looks like. So these are the number of the cells. These cells, there are the protrusions. The three-dimensional structure of it will be this. It is diagrammatic version where you will find on the surface of the spherical ball, or just like a head of a pin, where you will find the number of the flagella all along it, and there are the number of the cells which are together joined by means of cytoplasmic strands, and these are daughter colonies which are there for the modes of the reproduction is a sexual type of reproduction also. And this is how Ponderina looks like as a colony, as a coenobium, surrounded by means of an external gelatinous matrix. This is how Ponderina looks like. These are its flagella on the outer side, and this is also a gonium, which is externally covered by means of a gelatinous matrix. So these are the colonial types of the algae and their structures and their figures. Now there are non-motile Coenobial colony. Non-motile Coenobial is also called the cookoid form. Colony has definite number of cells. Lack flagella, closely attached together in a specific manner. Coenobium remains non-motile. And it is you have an example here, a net-like structure. See, this is a net, just like a fish net. It is hydrodictyon. And there can be also synedesmus. Hydrodictyon consists of many cylindrical cells. Here is a figure. This is one cell. This is another cell. This is another cell. They are end to end connected. And from this there is one more cell. So it is continuing with another hexagonal area. So they are joined end to end like a hexagon. And colony appears to be a network type, but it is not moving. That's why we call it as a non-motile Coenobial. Means number of cells are present there, but they are not moving. That's why called as a cookoid types of the forms. As you see in case of synedesmus, you have cells here, cells which are connected together. These are the processes at their end cells. This is acting as a Coenobium with definite number of cells, but is non-motile. Proceeding ahead, these are the figures of this is the volvox, as we have already discussed. This is the Ponderina, how it appears, and this is the Synedesmus. This is the Hydrodiction, which is just looking like an hexagonal mesh. So there are also aggregated forms. So aggregated forms or aggregation is there. Cells are aggregated irregularly. There is no regularity. The network is also aggregation, but that is in a particular irregular network. Irregularity is here, showing colonial mass of various size and shape. It's of three types: palmuloid, dendroid, and rhizopodial. Let us discuss them one by one. Palmuloid form is an algal colony with non-motile cells embedded in an amorphous gelatinous matrix. Gelatinous matrix may be secreted by protoplasm of individual cells. That is, the same cells are producing gelatinous matrix. A colony has an indefinite shape and size, not definite shape and size. No fixed number of cells. All cells are independent in function. A temporary stage. The polymela is also a temporary stage during asexual reproduction in Chlamydomonas, as is a permanent structure in Tetraspora. So we have seen polymeloid is the aggregation of the cells. This is a polymela stage in Chlamydomonas. This is how tetra, four cells together, how the aggregation looks like. They are of different types. Chloroscoccus, and then these are the Phaeocystis. These are some of the aggregates of the cells. And then we have a dendroid form. Dendroid form is also a non-motile colony, having a variable number of cells, but cell aggregates in a branched pattern. You will feel it is a tree-like structure under microscope. Plant body appears as a tree-like structure, looks unique. Mucilage often seen at the base of each cell, and cells are attached with the attachment. Cell have attachment with the substratum. You have chrysodendron, you have ecbelocystis, and dinobryon, and there is also Mescococcus, and these are some of the examples of the dendroid forms. So you have a tree-like structure which you see under microscope. Here you will find this is one cell, it's stuck to it. So these are, and here you will find the cells like this. They are stuck each other in an irregular fashion, looking like a tree. And this is how ecabellocystosis also appears, as has been quoted in the algae base. So there are other forms which are moving by means of their rhizopodia. Cells are united. This is one cell, another cell, and cell, but they are connected together, and they are moving with the help of their rhizopodias. So aggregation is there through connections, and then they are moving with the help of the rhizopodia. We call them as rhizopodial forms. Example is Chrysidiastrum, which is as you have seen the structure of it is appearing this, but they are connected by means of the plasmodesmatic connections. Now this was about the simple single cells, whether motile or non-motile. Now you have a second range where you have a filamentous type of a thallus. That means the thallus appears filamentous. Thallus with indefinite length. Cells attached end to end in a linear fashion. That is, you have one cell, second cell, third cell, fourth cell, this way. Filament is multicellular thread-like, formed due to repeated division of a non-motile cells. Division is in a single plane. That is why it forms a thread-like structure. And it is in a unidirectional. Division is unidirectional. That is why the structure of the thallus appears thread-like, not multidirectional. So we have a types of filaments. Filamentous form may be unbranched, simple branched, or falsely branched. Let us discuss them. Unbranched filament. Simple unbranched filaments are found in algal forms, which are free floating types. You have unbranched filaments, which are free floating. Free floating unbranched filaments have all cells alike, no differentiation into basal and apical cells. So it appears as a filament. There is no polarity in the filament, no base and the apical portion, as we have a Spirogyra or the Zygnema. But there are forms which are attached to the substratum by means of modified rhizopodial cells, and that is filament is differentiated into a base and apex. We have an oedogonium, we have a eulothrix. So these are the filaments algae. As we have seen, a Spirogyra is a filament from top to bottom same, having this type of a chloroplast, ribbon-like chloroplast, and the Zygnema is also a filament. Eulothrix and oedogonium, they are different. They have an apical portion and the basal portion. With the basal portion, that's the rhizopodial portion with which it is attached with the substratum, and oedogonium has also the apical and the basal portion. So these are the filaments which are of two types. One can be a simple, another can have polarity. As you will see in a structure here, it is an unbranched filament having cells end to end and with different type of the chloroplast in the nucleus, and Spirogyra also. Spirogyra filament, you can have a eulothrix where the basal holdfast is present there. It has a rhizopodial cell, it has an apical cell. So you, in case of oedogonium, you have a rhizopodial cell and you have an apical cell. So Zygnema is also all about having the same, no polarity is there. So this is on the basis of their polarity or non-polarity. But there can be branched also. They are simple, they are not branched. There can be simple but branched filaments also. Algal forms with branched filament. True branching and false branching. So here comes the two. One is the true branching, another is the false branching. True branching is branched thallus with a lateral growth of a main filament. That main filament will have a growth on the side, will produce another cell. Lateral branches arise as a result of occasional cell division in a second plane, as we will see in case of cladophora. Suppose you have a cladophora, it appears like this. Now this is the main axis. This is the main. At its lateral side there is one more division that is in the second plane that forms the branch of it. So one is the main filament and then are the branches on its side. As the cladophora appears, as a seaweed appears like this. When you see the branching, it has a branching pattern. So to some extent a true branching, because there is a different plan of the division here. It's in case of cladophora, and which is called as the branched filamentous algae. Then coming to false branching, filament appear branched. Actually, two separate filaments come close to each other, remain attached to each other by mucilaginous sheath, and branch does not arise as a lateral growth, outgrowth. So there is no lateral outgrowth, but just looks like a branch. For suppose a trichome of a blue-green algae may break either due to death or decay of the intercalary cells, and broken ends emerge out of the mucilage sheath as a branch. As in case of Cytonema, you see this is how it appears. This is no branching. This is basically how it comes out here. This is the main, but is not a branch. This is a true branching. Here it is prominent. So this is basically externally looking like as a branch, but this is a main branch which has shown a side twist, and that's why we call it as a false branching. Then comes the heterotrichous forms. Hetero means different. Trichous means a filament. It has a different type of a filament. That is, you will find a prostate area as well as an erect system in the filament, in the thallus. So thallus will show division of labor. That means it will be a prostate system attached to the substratum, and with the help of rhizopodial filament, as we have already mentioned, and erect system which will be above the prostate system is developed from the prostate system basically. So we have an examples here, ectocorpus, fistula is here, coleochete is here. These are some of the examples where you find this heterotrichous type of a habit. So ectocorpus is this, it has a prostate system, and from that comes the erect system. It is this erect system which bears then the sporangium, which is meant for the reproduction of this algae. This is the different types of this is the prostate system, this is the erect system, and this is how it appears. This ectocorpus is appearing like there. So is a brown algae, or as is seen in case of this is the prostate system, and the erect system in case of fistula, which was discovered by the Iyengar also. Then there can be different forms, as there can be an progressive elimination of the prostate system. It will have a perfect this erect system. Prostate system is slightly eliminated. Example, these are the two examples where you will find, while in case in case of coleochete, erect system is reduced there, and that has a highly developed prostate system, as you will see in case of the coleochete. This is how it appears. In this case, there is no prostate system but only erect system. In this case, there is no erect system, it is only a prostate system. So here is a prostate system, erect system, and there is elimination of the prostate system. Here it is the elimination of the erect system. It's all the prostate system which appears in case of the coleochaetes. So then coming to the siphonaceous type of the forms, where the thallus is unicellular, but it is elongated, and it has a multinucleate tubular non-septate. A unique algae where you find a cell is single cell, that cell is elongated, it has number of the nuclei, and it has a tubular like structure, and we call it as a cenocyte. Cenocyte is means many nuclei present in a common protoplasm. How we will have in one single cell number of these nuclei? That is only possible when a cell has divided multiple number of times, that its nucleus has divided, karyokinesis has taken place, but there is no septation, no cytokinesis. So growth takes place without cross-wall formation. That's why it looks like a cenocyte has a number of nuclei there, and it is a siphon-like structure, maybe simply branched. Example is in case of the Vaucheria, where we have this structure. These are a siphonaceous type of the algae, where you will find the central main hollow structure, and then this appears like this. This is how Vaucheria appears. These are its sex organs which are arising from the lateral sides. So you will find number of the nuclei which are there, and that's why we call it as the multinucleate. So the structure is called as the cenocyte also. Then also there is a modification as in case of the Botryddium, where you will find thallus elaborately developed. Clear division of labor. That is, you will find here a portion which is below the substratum and the aerial part also, but differentiated into aerial and subterranean portion. But here thallus is calcified and provide mechanical support. Here, so it has a calcification here, but rest it has a prostate system, it has an aerial system also, as is seen in case of the Botryddium. So these are some of the examples of filamentous and heterotrichous types of the forms. Now we have thallus which looks a parenchymatous. What we mean by the parenchymatous? We have already seen in case of tissues. A parenchyma is a tissue composed of thin-walled, closely associated cells which arise by the division of a common parent cell. That is, one cell has divided, repeatedly divided, and has produced a number of the cells. They have thin-walled cells. We call them as parenchyma cells. They can be storage cells also. So a modified filamentous habit. It is a multicellular thallus. Cell division occurs in multidirectional planes. In filamentous, it was in one plane. Here it is a modified filamentous habit. It's in multidirectional planes. And then there is also the septation or the cross-wall formation. Results in the formation of a thallus that is called as the parenchymatous thallus. Thallus may appear flat leaf-like. As you will see, division occur in one plane, ulva, laminaria, and fucus. So these are macroscopic types of the seaweeds, where you will find just like a leaf-like structure is found there. But that is because that's all parenchymatous tissue. So ulva appears like this, fucus appears like this, and laminaria appears like this. So this is their diagrammatic, how they appear. It is a flat leaf-like thallus. This is the central, the stipe, and then you have also in case of the fucus, where you have the hold fast because they are attached with the substratum, and then there is a thallus. So we will find there on the basis of the growth, little bit of differences in the growth, whether it's apical growth or uniaxial or multi-axial growth. Laminaria is a brown algae here, marine brown algae, where you will find the stipe and you will find the lamina here. So this is how it appears, and this is how the laminaria digitata appears. So this is a parenchymatous type of a cell where tubular parenchymatous thallus can be seen in case of Enteromorpha or Secatosiphon. Division occurs in more than two planes. Forms a complex thallus structure in codium and sargassum. These are the variants of the parenchymatous tissues. So cells of thallus are differentiated into central medulla, middle cortex, and the outer cells, as you will see in case of sargassum. Or algae even possesses sieve tube-like structure, not sieve, because I have already said the vascular system is not present there. These are not highly evolved organisms that way. So as it is similar to higher plants, you will find in microcystis a sieve tube-like elements. So this is how Cystosiphon appears, elongated structure here. Sargassum is appearing here. It has these air bladders and receptacles are there. Then there is the primary branches are there. Stipe is there, and then there is a holdfast also. Acodium, which is highly advanced, looks like this. Enteromorpha appears like this. So these are having a parenchymatous with some modifications even in the parenchymatous. These are the algae with parenchymatous types of the. So how is the growth taking place in the parenchymatous thalli? Growth of parenchymatous thalli may be diffused, intercalary. Diffused is all cells can divide. It will form a uniform structure. Intercalary means dividing region remains in the intercalary position, middle of the organ. Example is laminaria. Trichothallic is growth by specialized intercalary meristem located at the base of the terminal hair, as in case of porphyra. Apical is one or more well-defined apical cells divide, produce the remainder of the thallus, as you seen in the fucus. So growth, when we will discuss the individual genus, you will find these growth patterns in fucus, in laminaria and others, how they are taking place there. But a passing reference is given here. Pseudoparenchymatous forms. Now they are false parenchymatous. As the name signifies, they look like parenchymatous. They are not true parenchymatous forms. Thallus is formed with the close association of cells or filaments. In parenchymatous, what you will find? You will find the number of the cells. They will be same, but they are formed from a single cell, the division of a single cell. Here you will find number of the cells, but they are not formed from a single cell. They are the association of the cells or the filaments. That's why we call them as pseudoparenchymatous forms. Many axial filaments are gathered together in pseudoparenchymatous forms, forming an axis-like structure. It is basically a secondary development. So is a combination, a close association of cells due to interweaving of the filamentous. So we can say pseudoparenchymatous forms are formed because when the different filaments are coming together and giving an appearance of a multicellular parenchymatous like look, but they are not. So it can have a uniaxial axis form where filament formed by a single apical cell. Apical cell cuts off cells parallel to the base. Now, uniaxial, as you know, if there is a one organism, it divides at the tip, leaves the cells behind. They will be parallel to the base and has a single colorless central axis. So, because of one apical cell, there will be the central axis. But on the sides of the central axis, thallus formed by branches of a single filament. Thallus has one main parenchymatous axis and all other side branches which are formed from it. So we can say uniaxial is close juxtaposition of branch systems of a single main axial thread which form the thallus, as is given by the Fridge in 1935, where definition of the uniaxial axis forms, and it is seen in case of Batrachospermum. Batrachospermum appears like this, which has branches of limited growth, branches of unlimited growth, and the main thallus appears like this. And when you see the thallus, you find as if number of the cells are there. It seems a multicellular, and there are this whole structure is called as the glomerule. We'll discuss it when we discuss the genus, but externally it appears like the beads of thread or the beads, just like beads. This is one bead, and these are called as the glomerule, and is a macroscopic type of an algae. And you see how the growth is taking place. So it has an apical cell. Apical cell forms downward the cells. These cells will grow, and they will form on sides pericentral projections. They will have division in this plane, and they will grow the cells, and there will be some more divisions on this side. They will cover it. These cells will cover it. What they will form? They will form the cortical cells. But main axis is one. So that is formed from the single cell. That's why we call it as a uniaxial. And there can be then secondary cortical cells. But when you take a cross section of it, what you will find? You will find a central cell, and you will find it is surrounded by means of the number of the cells. But those cells are also formed from the single cell. They will be lateral branches, which will be in the form of branches of limited growth and branches of unlimited. Unlimited growth will be this, and the branches of limited growth will be this. So it is in case of Batrachospermum, so you have an uniaxial type. So growth is taking place in one plane, in an apical cell, and which is parallel to the base, and so it will have a main central axis. So we have also in multi-axial form. Multi-axial form is basically main axis is formed by association of many threads. So here it is number of threads together, appearing as more than one axis. It appears as more than one axis. Internal filaments by close association. They make a solid core-like structure. So here we have number of threads together. So secondary external filamentous that forms the cortication may cause increase in the thickness of primary thallus, as is in case of Polysiphonia, Chondrus, and Desmertia, which is also called as the acid weed. A very interesting weed which is an annual or a perennial, but it stores sulfuric acid in intercellular vacuoles. But once it is exposed to air and a suicide mission, they release the acid, thereby destroying themselves and the nearby weeds. That is also having a multi-axial form, and multi-axial form is formed by means of association of many threads together. It is how Polysiphonia appears. So these appears as different multicellularity, but these are the different types of the filaments which are together, and they are appearing externally as if they have a multi-axial growth, but that is because the different types of the filaments are together. These are the diagrams of the Polysiphonia, and this is how multi-axial growth is taking place in these type. And you will see apical cell is one, then it has a number of these filaments which are coming together, and once you take a section of this, what you find? You find a central siphon, you find pericentral. This whole is looking as a core, and then you find external cortication, you will find more cortication, but they are not basically the well organized have not the well organized anatomy. It's not that they have divided in different planes. Basically they are the different filaments which are together. That's why it appears as multi-axial. So it is a Desmertia ligulata, which I called as the acid weed. This is how dry it appears. This is the Desmertia, and this is when you take a section of it, you will find the central cells surrounded by means of these, and then the cortical cells. But mind you, these are not basically as is done in case of the higher plants. So anatomy of them is multicellularity, but that is not the differentiation has not taken place there. That's why I said they lack what you called as the this conducting or the vascular tissue. So this was basically about the range of the thallus, and in case of the algae, as you have seen, range of thallus structures in algae is vast, with forms range from simple single-celled to structures to complex multicellular structures. And these play a crucial role and a critical roles in the survival and the reproduction of the algae. And this was a broad visit to different types of the fascinating organisms, especially to see the range of the thallus structure in these algae. Thank you very much for the patient listening. Hope you have understood it, and be safe and be happy till the next video is uploaded. Thank you very much.